Now, there was some local research that was published recently that was a little concerning, and I really wanted to share it with you. Joining me now is the lead researcher of a newly published study in the journal Applied and Environmental Microbiology. Her name is Suzanne Young. Suzanne's a PhD student at the University of South Florida. And in her study, she found dangerous antibiotic-resistant bacteria in the environmental waters of Tampa Bay following a sewage spill. And these are not normally found outside of a hospital setting. So, hello, Suzanne, and welcome to the show.
Hi, thanks for having me.
Yes, ma'am. I appreciate you being on.
No problem.
Let's go ahead and start with the scenario in case people don't know about what happened in 2014 in St. Petersburg. Can you go ahead and lay out the groundwork for that, please?
Yeah, sure. So this was a sewer line break in Pinellas County that happened in September of 2014, and we actually learned about it through local news and decided to go out and sample the sewage and the water and soil that were being impacted, or that we suspected were being impacted by the sewage. It ended up flowing for about 3 days before they could fully repair it, and about 500,000 gallons of sewage were released during this spill. And most of it was kind of diverted down this drainage ditch, which was where my sampling took place.
And what waters did the sewage end up going into?
So the direction of that flow was towards in the Pinellas Park area, towards Long Bayou and Cross Bayou, and then further away in Boca Ciega Bay.
Right.
But we didn't detect the vancomycin resistant enterococci or the resistance gene that we were targeting in those recreational waters. So that was kind of the good news, is that we couldn't see the signal of the spill that far out into the bay. Right.
Now, you already talked about the organism that you detected, which is the troubling thing. Can you talk briefly about vancomycin resistant enterococci, and where do we normally find this?
So VRE, as they're commonly called, are one of the most common hospital-acquired infections. So we hear a lot about MRSA and C. difficile as infections that you get in the hospital, and VRE is one of those. So it's commonly found in hospitals and in hospital sewage when you directly sample the waste coming out of the hospital. So we were concerned because in the past we haven't found VRE that's resistant at this level, or the resistance gene that carries vancomycin resistance in other sewage, in residential or domestic sewage outside of a hospital. So that was where the concern was, in seeing what we think is this spread out into the community.
Sure. Now, there's a lot of different pathogenic bacteria that are found in sewage, but this particular strain was a little bit more significant because of a gene. Can you talk about the vancomycin? I think it's the vanA gene.
Yeah, the vanA gene is the most concerning as far as public health and human health. It carries this high level resistance, so a lot of enterococci, which are natural in the human gut and in the environment,
right.
And some enterococci can carry an intrinsic or inherent resistance to vancomycin, but the vanA resistance that we found is a high level of resistance, meaning it can tolerate a lot higher concentration of the antibiotic, and more concerning because the gene is carried on these mobile genetic elements that can transfer between bacteria, between pathogens.
Yeah, so that's a huge problem. So, I mean, vancomycin is essentially a last resort antibiotic for gram-positive organisms like
right.
the staph and streps. And having this ability to transfer this gene to Staph aureus and other types of staphylococci makes something like MRSA even that much more worrisome.
Right. So we have a few cases. It's very rare so far, but in the past couple of years, there have been about a dozen cases of what is called VRSA, or vancomycin-resistant staph.
Right.
So that, although it is pretty low in incidence, it's just something we need to watch for and
Sure.
be careful of.
Sure. Now, what was the specific organism? I imagine it was Enterococcus faecium, right?
Yes.
Yeah. Something that's seen in a hospital from time to time, and it's typically considered vancomycin resistant.
Yeah.
Suzanne, how long was the VRE persistent in the water and the soil that you tested?
So we were able to detect this vanA gene up to 12 days after that initial break in both the water and the soil that we sampled.
And does that tell us anything? Does that mean it's not normally found there? It was strictly from the sewage?
Right.
Yeah,
Yeah, that's what we inferred from our 7 weeks of sampling, because we only detected that gene within the first 2 weeks after that sewage spill.
Okay, great.
And because we didn't detect it either by culturing or by targeting this resistance gene, we could pretty confidently conclude that it was not there naturally in the environment.
Sure. Now, what other public health concerns did your study raise?
So in this case, the risk to the public was not so great, because people weren't really flocking to that drainage ditch to go swim in it.
Sure.
But when these things happen in different parts of Tampa Bay at popular beaches, or when it's an even greater volume being released, it's more concerning as far as exposure to the public and the potential for people to get an infection that's not easily treated with antibiotics. And it is not easy to get an infection or a VRE infection if you're a healthy adult, but if you're part of the very young or elderly population or immunocompromised, you're at a greater risk to get an infection from the water.
Sure. Now, I read in a press release a quote from you. You were talking about your concerns with antibiotic use, and you mentioned animals. Can you talk about that at all?
Yeah, so it's kind of a corollary to this. It's not directly related,
Right.
At least with this sewage spill, but part of the broader problem of antibiotic resistance is the amount of antibiotics that are used in animal agriculture. And we've seen with the case of VRE, we've seen this exchange kind of between animal husbandry and using a vancomycin type antibiotic in those animals, and then seeing vancomycin resistant infections in humans that they could genetically link to the use of the antibiotic in animals.
Yeah.
And while we don't use that antibiotic, it's called avoparsin. We don't use that in the U.S., but it doesn't mean there aren't similar patterns that could appear.
Right. Now, are there any final lessons from your study, and where do you go from here? Do you continue with this, or is this going to be the end of it?
Yeah, so we'd like to keep sampling just sewage in general, and when sewage spills happen, to see if we're finding the VRE and the resistance gene in sewage consistently, and over a broad geographic area. There is one other study in Washington State where VRE have been detected in domestic sewage. So I think getting a better idea of how much is out there, where it is, and whether this is something that's just in our sewage now. So this kind of environmental surveillance. And then also some projects we're working on in the lab with how these bacteria can survive in the environment, how they survive under different environmental conditions, and whether maybe there are some conditions that can hinder their survival, so we can try to mitigate the potential effects of releasing these antibiotic-resistant organisms and their associated resistance genes into our waterways.
Oh, very good. It sounds like you're doing some really important stuff up there at USF. Thank you, Suzanne Young, for your time and expertise, and congratulations on the publication.
Yeah, thank you.
You're more than welcome. Thank you.